Hello, my name is Jenna Mulreany, and I am a third-year undergraduate student at Duquesne University. I have been working with PhD student Michael Cardos and Dr. Sarah Woodley on our research of using PS6 immunoreactivity as a marker of neuronal activity in the context of salamander reproductive behavior. What originally sparked our interest for this work was the current lack of knowledge of brain activity in salamanders during the courting and mating processes. We first considered what we initially know about pheromonal communication during the mating process of salamanders and how chemical signals are exchanged between the male and female. PS6 immunohistochemistry is a new method to track neural activity that has only been performed a few times within the past few years. PS6 is a ribosomal protein that can be marked through staining during neuron activity in response to a wide variety of stimuli when the protein is phosphorylated. The neuron cells that we are targeting to mark will have experience and action potential that produces antibodies that we can stain in recognition of the PS6 protein. Based off of the experimental design I set up that I will soon further explain, I predict that there will be more immunoreactive PS6 neurons in the brains of salamanders that have recently mated compared to those that have not. When developing the experiment, we wanted to design testing chambers that would best represent a normal mating experience for the salamanders as if they were in the field. The experiment was performed in the evening with minimal light on the salamanders, and they were placed in clear testing chambers and monitored for mating every 15 minutes. There were 6 pairs that were allowed to complete the mating process, 4 pairs that were separated before the mating process had been complete, 4 males that were handled to induce a stress response for a positive control, 8 males left alone as controls, and 3 females that were also left alone as controls. The positive control was from previous research performed by Zoe Brown of this method, with stress being the induced stimuli as opposed to mating. The mating process was scored based off of observation of which part of the courting process and mating process the pairs were at. At a score of 0, the male had shown no interest in the female. At a score of 1, the male had begun approaching the female. At a score of 2, the male had begun touching or rubbing the female. At a score of 3, the pair had begun the tail straddling walk, which is when the female climbs onto the back of the male and he begins to guide her to where he will be depositing his spermatophore. And at a score of 4, the male had deposited his spermatophore, and we could visibly see it in the testing chamber. As we only observed the behavior every 15 minutes, the spermatophore had been inseminated by the female in most of the mating cases, and we could visibly see this inside the cloaca of the female after removing them from the testing chambers. 2 hours after the mating process had been completed, the salamanders were euthanized and their tissues were preserved for sectioning using a cryostat. After the tissue had been cut and fixed to microscope slides, the immunohistochemistry steps were then ready to be performed. The key steps of the immunohistochemistry method was the addition of the ultrasensitive ABC peroxidase staining that was what targeted the primary and secondary antibodies within the neuronal tissue. The ABC came in a kit with 2 reagents, avidin and biotinylated HRP, and when these reagents bind together, they create a more complex molecule that can target the primary and secondary antibodies together, creating an enzyme substrate that helps to amplify the activated neuronal signals within the tissue. A DAB stain is then added, which is what binds to the amplified enzyme substrate to stain it for visible imaging. I have recently finished the immunohistochemistry process, but have not yet been able to image my own tissue. From previous research done by Zoe Brown with using the PS6 immunohistochemistry method, with stress being the induced stimuli, I have included her immunohistochemistry results to display what I believe will be similar results to the staining on my own tissue. I plan on imaging and counting my activated cells by hand or with the ImageJ software within the upcoming weeks. As seen in the expected IHC results section of my poster, you can clearly see how the stain from the immunohistochemistry is distinctly visual as opposed to the control tissue without any induced stimuli when using the PS6 method. Along with observing the activated neurons in the tissue from the different treatment groups I designed, I will also be looking into what regions of the brain or spinal cord have the highest amount of activated neurons because of the performed stimuli. Moving forward, I predict that there will be higher amounts of activated cells in the telencephalon and amygdala regions of the brains, as those areas are directly linked to the vomeronasal and olfactory systems, which are both major contributing complexes in the salamander mating processes. As that is all I have now, I would like to thank the Woodley Lab and the Department of Biological Sciences at Duquesne University for providing me the opportunity to learn and perform this research. I would also like to thank Zoe Brown for her images pictured in my poster as successful controls when moving forward with my own research. Thank you.